Human chromosome 2 is one that I really want to focus on right now. Human chromosome number 2. I want to show you why it's important. There are over 1,200 genes in this chromosome. All of them are important. But I'm going to talk about one. I'm just going to talk about one. It's called TBR1. Gene TBR1. Here I'm pointing up here and you can't even see this. TBR1 is responsible for the neocortex that gives us so many of our capabilities. The mirror neurons we're talking about, they're in our neocortex, the human neocortex. TBR1 is responsible for that. TBR1 is responsible. That neocortex, if we didn't have it, we would not have our uniquely human capacities of empathy and sympathy, compassion, the ability to self-regulate our biology. All comes because of a mutation that happened in chromosome 2. So you begin to see chromosome 2 is really important. I want you to see exactly what I'm talking about. What you see in the green on the right-hand side of your screen, that's the neocortex. That is a result of human chromosome number 2. I'm not saying we wouldn't be alive without that, but we wouldn't be who we are today without that. We wouldn't be here having this conversation. So you begin to see human chromosome 2 is really important. Would you agree with that? If it is responsible for our capabilities of sympathy, empathy, compassion, self-regulation of our biology, and many other things, where did it come from? This is where it gets really, really interesting. Discover number 15. See, we're going really fast. Here is the paper I was just talking about. This is from a peer-reviewed scientific journal from the Proceedings, National Academy of Sciences. The volume is called Genetics, volume 88. Very technical paper. The bottom line, what they're saying, is that our chromosome 2 is the result of a mysterious fusion of ancient DNA. It's the relic of an ancient DNA fusion. It happened in a way that cannot happen in nature. I want to show you what that is. Not only did DNA fuse in a very unique way to give us the DNA, after the fusion happened, genes were added, genes were taken away, and genes were fused to stabilize those chromosomes coming together. What does that sound like to you? Have you heard anything like that earlier today? Genes added, genes taken away to facilitate that function. Up until the time of CRISPR, we had no idea how that could ever happen. And now CRISPR technology shows us exactly how it can happen. And I want you to see it. Here it is right here. This is your human chromosome number 2. And what I want you to see is this pinch point right there. That is where two pre-existing, they're called ancestral chromosomes, have been fused into one long, this is the second longest chromosome in your genome. Fused together, and genes were added and taken away to give us these capabilities. Now, we're going to talk about telomeres tomorrow. Many of you know, if you've been with Joe Dispenza, you know about telomeres. They only are found on the end of chromosomes, on the end of the DNA, right there where that pink is. It's the only place they're found, except human chromosome 2, they are in the middle where they were fused together. Here's what I mean by it. And if you're a biologist, I want to be very honest. Chromosomes can fuse together in nature, but not the way ours did. And here's what I mean by that. Okay, here's a couple chromosomes. They will break sometimes. Toxins in the environment, ultraviolet light from the sun, chemicals. So environmental things can break the chromosomes. That's true. But here's what happens. When those chromosomes will break, typically, they'll break. Oh, I wish this pointer would work here. Okay, I have to stand right here. When they break, what will happen is part of the chromosomes are absorbed, and the remaining pieces will fuse at that from the break. So here's the break right here, and now they're fusing together. Can you see that happening? It's not what happened with you and me. Here's what happened with us. So if you have that happen, you've got that fused chromosome right there, but there's still telomeres on the ends. Here's what happened with us. Check this out. The telomeres are still on the ends. With us, these are chromosomes. Okay, there's a chromosome on top with telomeres on the end. So the blue is the telomere. So there's one chromosome on top, and the blue telomeres on the top and the bottom. Another chromosome on the bottom, blue on the top and the bottom. Those two were fused, and where did the telomeres end up? Where do they end up? You tell me. Exactly, in the middle. Telomeres don't belong in the middle of a chromosome. This is how we know that these were fused together. This doesn't happen in nature. It doesn't happen. And if that's too complex, there's a little movie to help understand this. Here's two strands of DNA, two chromosomes. Genes are added, genes are taken away, and the result is fused together into one new, shiny, new human chromosome number 2. Sympathy, empathy, compassion, self-regulation. Right here. The timing, the fact that this happened exactly when humans emerged 200,000 years ago. The precision with which the genes were added and taken away. And the end result, us, all suggest something beyond evolution. This is a problem for scientists. It's called forbidden territory. And I love forbidden territory because it suggests that the mutations were done intentionally. Science cannot accommodate intentionality when it comes to our origin. They're not prepared for that. They don't want to be. They don't want to follow this evidence. If there is an intentionality, it implies a purpose. What is that purpose? I think you and I are exploring it. To fully realize what it means to be human, to self-regulate this mechanism, this conscious vehicle in a healthy way. Bring joy into our lives and bring peace onto the planet. That's how we do this. Not living in fear, but living in the confidence and the trust of who we are and our relationship to our bodies and the world. Just for your information, if you say, well, Greg, maybe it's pretty unusual. Maybe it could have happened. The odds of that DNA forming the way it has, I love this. This is from British astrophysicist. His name is Fred Hoyle. He says the odds of this happening are the equivalent of a tornado sweeping through a junkyard, and when the tornado leaves, behind is a Boeing 747. We've talked about this twice today, so I want you to know this is what they look like. And for a non-mathematician, what does that mean? Right there, there's a Boeing 747, big planes. That's not going to happen. You're never going to see a tornado go through a junkyard and even leave a part of it. You won't see a wing form from that junk. You won't see the fuselage. It's not going to happen. So what they're saying is the odds are impossible. I'll just tell you, any mathematicians in here? Anybody like math and engineering? Okay, so I'll tell you, mathematically, If the odds of something happening are 1 in 10 to the 400th, that is called impossible. If you get a lottery ticket and the numbers are 1 in 10 to the 400th, forget it. It's impossible. So that's impossible. The odds of our DNA forming the way it has, the odds of that happening are 1 in 10 to the 600th. It's more than impossible that we are the product of a random process. More than impossible. So the question is, who or what is responsible? Well, you can watch a lot of Gaia content, have a lot of ideas about what that might be. What I'll say to you is this, as a scientist, I want to be very honest with you. It depends on who you ask. Because every ancient and indigenous tradition that I've studied, every one, bar none, all have an answer to this question. And none of them are the product of random processes. None of them. They all have a common theme. And they say the Sumerians, they have their story. All right? The Aboriginals, they have their story. You can see giving birth to the human species right there. The Hindu traditions, they have their story. Christians have a story, and they got great pictures with the Christian story about how this happened. All right? Native indigenous, the Navajo, right down the road here in Four Corners. Scientists have had their story, but their story has broken down. If you watch television, there's a story right there. But now the evidence is changing everything because the evidence is giving us a new story.
This session's presentation focuses on the role of telomeres, the body's equivalent of the fountain of youth. We are all mortal organisms, which means we will inevitably die. When our time comes, does it really mean death is an ending to our existence? An assessment of our cell membrane's population of protein receptors suggests that a part of us survives after death, an energy that implies the existence of a spiritual realm. Animals and plants have a generally defined lifespan between the time they're born and the time they die. When the science of genetics was developed, the first thing geneticists were looking for were to identify the genes that presumably control our aging. But is aging a genetic character? In the early 1970s, Russian theorist Alexei Olovnikov first recognized that the ends of a chromosome could not be replicated when the cell divided, a finding that revealed a segment of the terminal DNA sequences in a chromosome are lost every time a cell replicates. When the loss of the DNA strand reaches a critical level, the cell can no longer replicate, and then it dies. Further research revealed that there are extensions of non-coding DNA at the terminal ends of the chromosome. These extensions are called telomeres, and they have two fundamental functions. One, they prevent the DNA double helix from unwinding, an action that will cause it to be degraded by enzymes. And two, the telomeres allow the DNA to be copied without losing genetic information. The DNA copying enzyme, DNA polymerase, is what duplicates DNA molecules. However, physical limitations prevent it from reading and copying a short stretch of the DNA at the terminal end of the DNA strand. The reason for shortening of the telomeres is very simple. Imagine a DNA molecule as a railroad track, and that the enzyme that copies DNA, the DNA polymerase, is an engine moving down the track. As the DNA polymerase moves down the track, it leaves behind in its trail a duplicate copy of the DNA on which the engine was riding. However, the enzyme cannot copy the DNA piece on which it is sitting. So, when the DNA is being copied, the polymerase moves down the track, copies everything, but at the very end of the strand, the engine of the polymerase is sitting on that track and cannot copy it. Significance is profound, because it means that every time you copy the DNA, it gets a little shorter than the original version. So every replication of DNA reduces the length of that DNA molecule. Well, this is what leads to aging, depression, and disease. So nature facilitates us in the ability to extend the replication time by adding a piece of DNA at the end of the DNA strand that doesn't code for anything. It's just an extra piece of DNA track, so to speak. A DNA is a double helix, and it is very stable as a double helix. But if the DNA starts to unfold and the helix starts to separate, then the free ends of the DNA are now opened to digestion by enzymes. So if a DNA unfolds, enzymes will actually destroy the DNA molecule. So it turns out that the telomeres, the little extensions at the end of the DNA, act as a mechanism to prevent the unwinding of the DNA. To give you an analogy, when you have a pair of shoelaces, you also note at the end of the shoelace is a little plastic capsule at the very end. It actually has a name called an aglet. Aglet holds the strands together to prevent them from unwinding. Because if you lose that aglet, what happens? The shoelace starts to unwind and you get a big bushy end to that shoelace. It's very difficult now to push that shoelace through the eyelet in the shoe because it's all flared out. And so it actually decreases the function of that shoelace. Well, simply the same thing occurs in DNA. If the DNA helix unfolds, then the DNA is going to be broken down by enzymes. But the telomeres act as the equivalent of an aglet. They hold the strands of the DNA together to prevent its unwinding and subsequent degradation. So that's one primary function of the telomere. But the primary function of the telomere turns out that it makes an extension of the DNA that allows the DNA to be shortened every time the DNA is copied without affecting the gene programs. So this allows so many divisions before we run out of telomeres. And then it was believed that a lifespan of an organism is based on how many times a cell can divide before it loses those telomere extensions and then cuts into the DNA program. Leonard Hayflick, a scientist back in the 1960s, did a calculation based on how long the telomere extension was and how many times it could divide before the telomere was gone. He comes up with a number that a cell can divide about 60 times before it loses its telomere. Well, when he put that information into our biology, how many years does that account for? Well, Hayflick's number comes down to a human can live to approximately 90 years before the telomeres are lost and then subsequent replications of the DNA interfere with the gene programs. How interesting. It turns out we live about 90 years, and that's apparently how long the telomeres exist before being cut off. Well, that sort of said our lifespan as a human is limited. In 1984, research scientist Elizabeth Blackburn discovered an enzyme, telomerase, that can extend the length of telomeres, which when translated means the potential to extend the human lifespan. However, this enzyme can be activated or inhibited based on what we're experiencing in our life. So for example, things that will inhibit the enzyme and prevent us from extending our lifespan include things such as bad nutrition, childhood abuse, domestic violence, post-traumatic stress disorder, no self-love or loss of love, no purpose in life. Each one of these characteristics really takes away the character and quality of our life, which in a sense, if you think about it, means, well, I'm not happy with my life the way it is, so ending sooner is not a big problem. But in contrast, there are activities that enhance telomerase activity and can extend our lifespan and our health. And these activities include, number one, providing good nutrition, especially omega-3, exercise, happiness and gratitude, as well as a positive outlook, self-love and love, and having a purpose in life, meaning you have something you have to do. And this is really important because having a life purpose is really a conscious intention saying, I'm not finished, I have more to do, and that feedback enhances the telomerase activity to extend your life. In fact, healthcare givers have a longer life than other people that are not involved in that activity. So if you understand this and start to live with love and purpose and happiness and gratitude, then you're starting to make your life extended because you're going to activate this telomerase, increase the telomeres, and allow your stem cells to replicate more frequently and also then in the process extend your life and your health. Well, in addition to the telomerase, there was also another insight into aging, and this involved what is called parabiosis experiments. Now, parabiosis is when you take two organisms and surgically put them together so that the circulation system of one is connected to the circulation system of the other. And they do a lot of experiments with putting rats together or mice together and then trying to find out what is the consequence of sharing a circulatory system. Well, here's what they found. They found that when you put a younger animal and parabiose it with an older animal, that the older animals will actually start to express youthing. They slow down their aging and they improve their health and vitality when their circulation system carries blood from a younger organism. Well, now the significance is this. Is the mechanism that influences youthing present in the blood is something that enhances youth, like present in the young blood, or is it something that compromises youth by something that's released into the blood of older individuals? Well, that's not been worked out yet, but the conclusion is very clear. That younger blood enhances the vitality of older organisms, again suggesting that some component in blood is very much involved with the length of our lives. But the length of our lives inevitably will come to an end, and we'll finally age and we'll finally die. And the question is our existence fully terminated with death? In Newtonian terms, the physical realm that we live with, well, that dies, so the answer is yes, we're terminated. But in the realm of quantum physics, we're offered an alternative possibility, for the realm of energy is neither created nor destroyed. Insight into our potential spirituality is linked to a unique biological identity. No two people are biologically the same. And this is evidenced by the simple fact that we cannot exchange cells, tissues, or organs with one another. So if I put my cells into your body, your immune system will distinguish that it's not your cells. It distinguishes self from foreign cells. And why this is really important, it says that somewhere on our cells there's an identity that an immune system can recognize and distinguish self from not self. Well, to try and understand that, I'd like to take two identical cell types. Let's say two liver cells. I want two cells that are carrying out the exact same function. But there's something different between them. One of them is Bruce's cell, and the other is Margaret's cell. So now there are two identities, two liver cells, but each has a unique identity. The question is, where in that cell is the unique identity to be found? Now, first of all, I know the nuclei of the cells are different. In experiments that I've even done, you can remove the nucleus of a cell, and the cell can live for weeks without the nucleus in it. So in our little model that I'm going to show you, let's ignore the nucleus for the time being, and just look at the cell. Now here's the issue. When I remove the color from the two cells where they're exactly the same, guess what? The only color that's left in the image is on the surface of the membrane covering each cell. And I said, well, what is on the surface of the membrane that gives us identity? In our next episode, we're going to focus on fractal geometry, the mathematics governing